Deixa eu emocionar aqui.
Oi?
Ok, estamos ao vivo.
Bom dia, pessoal. Bem-vindos ao nosso segundo dia de Semana de Abertura do Programa de Pós-Graduação em Ecologia e Evolução da UERJ. Hoje teremos 4 palestras dos nossos pós-docs e professores visitantes. E vamos começar de manhã com os 2 pesquisadores que são bolsistas do projeto Cap Sprint. Então, o Thiago Souto e o Brian Jennings. Como eles são ligados ao projeto Cap Sprint, as palestras deles serão em inglês, mas não se preocupem que eles vão falar devagar e vai ser tranquilo, né? Qualquer dúvida, vocês depois podem fazer perguntas também em português, né, no final das palestras. De tarde, teremos as pesquisadoras Luciana Costa e Carla Siqueira. Tá? Então, vamos começar, vou apresentar o Thiago. O Thiago fez graduação na Unirio e o mestrado aqui no nosso Ppgee, orientado pela professora Mariana Vale. E ele fez o doutorado na Queen Mary University of London, né? E por isso ele voltou da Inglaterra e veio aqui para fazer um postdoc aqui com a gente. Então, a palestra dele, hoje o título da palestra é Interaction Network Models in Climate Change Scenario. Então, vou parar de compartilhar aqui a minha tela. Não sei se o Bruno quer falar alguma coisa também. Opa! Não, ok. Então, vou deixar a palavra ao Thiago. Pode compartilhar a sua...
Sim. Muito obrigado, Eugênia. É um prazer estar participando aqui desse evento e poder falar um pouco também da minha palestra. Vou mudar aqui para o inglês, né? Que nem a gente combinou. So, today I'll be talking a little bit about the research I've been conducting the past year as a postdoc. And the title of the talk is Interaction Network Models in Climate Change Scenario. And I'll start talking about a little bit about how I came to this idea, and then I'll walk you through the methods I'm using, and then I'll do a little bit of a discussion towards the end. So one of the things that always interested me was understanding where animals are and what kind of things they're doing in these places. And as I studied and went through my undergrad and everything, I found out that biologists have been doing this for a long time, and every time with more refined ways of doing that. So it's no surprise that when I started my master's here at the program at UERJ, I went to study the distribution of a bat, and I learned about the use of distribution models to a wide range of applications. And I found it amazing because it's a method that you just need a little bit of data, like the occurrence records of certain species and some bioclimatic variables, and you can do some very detailed models of their distribution. And one of the most amazing things that I found out at that time is also that with the projections of climate for the future, you can also project the distribution of these animals in the future, like in this picture here from Fisher et al. 2011, where you see the potential distribution of these 3 mosquitoes in Central Europe. And with the warming and the change of the climate, these areas are actually more suitable for these species. And and also another thing very interesting is that you can stack all these models, and you can also work with like the richness and how the species richness will change over time, and you can model that. And I think this is very interesting and important because, as we saw with the last IPCC panels reports, the threats from climate change are severe and they are already here and happening, and we need to find figure out ways of dealing with that and avoiding all those negative impacts, because it will impact not only the natural systems but also our human society. and and then when I went to Queen Mary to do my PhD, I ended up working with the other type of question: what are the animals doing in these places? And one of one ways of doing that, it's like tracking what they are eating. And biologists have been doing this for centuries, and in the last decades, with the use of network theory, you created a very robust mathematical framework to study these interactions between species. And here in these networks, you can see that species, the nodes represent species, and then interactions are represented by the links between these nodes. And one of the most interesting things of this is that you can study any type of interactions, ecological interactions, using this method. And with the advent of technology and public databases, the amount of information we have about species interactions, it's always increasing. This is a result from my PhD, where we did like a 4 trophic level network with the plants here on the bottom, bats that feed on these plants here in blue, and then the bat ectoparasites, and then the endosymbionts of the parasites. And this is just to show that the amount of information on species interactions nowadays, we can get like a very detailed picture of it. So after finishing my PhD and looking for what to do next, somehow I wanted to connect these dots of the things that I had studied, and I started thinking on how can we use the species distribution models to predict climate change impacts on ecological networks. So in this last year, I've been thinking a lot of how to do this, and I started trying to test it in some subjects. So the first thing that I had to decide was like what type of interaction I'll be looking at. And I decided to look into pollination, which is a mutualistic interaction, and it's a very important ecological service, both for food production and also for the whole vegetation cycle, because lots of species really depends on pollinators for their reproduction. And I had to decide a group of pollinators, and then I decided to study the nectar feeding bats that I had worked before in my past research. And then I decided to use also the hummingbirds, because it's a similar group in size, in body mass, but they have very different foraging habits. And where to test it, I decided to go for the Atlantic forest first, because that's where we live in, and also because of it being a hotspot of biodiversity and highly threatened as well. And also it's in the region of the country that it's heavily occupied, and we need to know how to protect this biome in face of all the climate changes that are coming. So I wanted to answer a few questions. The first of them, it's like if pollination networks are threatened by climate change, and we'll be looking especially on this process of distribution mismatch. That's when a pollinator, while tracking their suitable environment, the pollinators move to one place and plants to another area, and then they just don't occur together anymore and they can't interact anymore. And also, I'm interested in finding out where these networks are under threat. So we'll project these results in the geographical space so we can figure out where these networks are changing the most in the future. And then if bats, hummingbirds, and plants are under the same level of threat. So the first step in this method is you got to define the species you're using. I ended up with a list of 41 hummingbirds following Lima and Silveira in a list in the Aves da Mata Atlântica book, and with the 8 Glossophagidae bats, the nectar feeding bats that occur in the Atlantic forest. And from this list, I went on to search all the recorded interactions of these animals with plants, and I searched in a series of databases, public databases for interactions, and also in the literature. And in the beginning of this year came out this paper, Atlantic Pollination, a data set, and a lot of the information I'm using came actually from this data set. And with this, we end up with a list of possible interactions. And we had more than 2,400 interactions among 45 pollinators and 770 plants. A few of the pollinators just don't have any interaction recorded, any visitation to flowers or anything like this. So that's why we're not dealing with all the 49 pollinator species that I showed here before. So after that, after the list of interactions, we'll build an incidence matrix of possible interactions. This is a binary matrix where one represents a previously recorded interaction between a pollinator and a certain plant. This is just a subset of this matrix, which is huge, and we'll save it to use it later. And the next step, the next phase of the analysis is building the distribution models for each species. And then you need to search the occurrences. You have several public databases like GBIF and SPLink that you can search for these occurrences. And then you need to clean from all the duplicates and wrongly geo-referenced records. And then we also do some thinning. In our case, we used 5 kilometers, so you don't have more than one record inside the 5 kilometers radius. And from all these, we start building the models. And we're using the world clim bioclimatic variables for the present, and then for the future in 2080 under 2 climate change scenarios: the business as usual, RCP 4.5, and the worst case scenario, RCP 8.5. And we do a person correlation test. So we ended up with like these 3 bioclimatic variables, the least correlated bioclimatic variables for the area that we are studying. It's interesting because we have some very widespread species. So when considering the bioclimatic variables, we had to include also areas in North America and all that. And I think that's why we ended up just with a small set of variables as well. And with this information, with the records and the bioclimatic variables, we built an ensemble model using several algorithms available. And we end up with a distribution model, environmental suitability model for the present and for the two future scenarios. And with this, we'll divide our study area in a grid. Here, you can see the Atlantic forest divided in hexagons of 50,000 hectares of area. And for each of these hexagons, we gotta calculate the average suitability for each species. And that's where the step I'm currently working on. I'm finishing to calculate this for all the species so I can do the next phases of the analysis. So with all the average environmental suitability of all the species calculated, we'll end up with a new matrix where you have the product of every plant and pollinator in environmental suitability, the product between these two species, and you fill the table with it. Like you fill a matrix with the product of the environmental suitability of these two. And we use this product as a proxy for the interaction strength, as we know that also the environmental suitability is correlated with species abundance. So in order to use a proxy for this, the interaction strength between these two species, we multiply both environmental suitability. And then we end up with that first matrix. This is how we build the matrix for our network. We use the first matrix that we did with the possible interactions, and then the second matrix where you have the product between the average suitability of each pollinator and plants occurring in that area, and then we multiply one matrix with the other, so we end up with our final network. Because some pollinators or plants may not be in a certain area, and also they may be together, but they may not interact. They may not have any interaction recorded before. So we end up with this matrix so we can build a network for each of the hexagons. And then with those networks, we'll calculate a few metrics like connectance, degree distribution, nestedness, modularity, and we'll plot these metrics in space, and then we'll just calculate how much these network metrics are changing between the future and the present. So we'll end up with a rate of change in the future for these metrics in the space. And this is just a representation of all the possible interactions. It's a bit messy. We can't even read the plants names here in the bottom, and we have the pollinators here. Because this is a binary network, also the width of the bars are related to the degree of each nodes. So basically how many species each node interacts on the other level. And looking at the larger ones, we can see that probably these are the ones that are keeping most of the interactions in the system. and probably they are also more generalistic in their environmental requirements, and most likely they'll be the ones surviving and maintaining some of the function in the ecosystem. While the smaller bars, they interact with very few species, and probably they'll be more at risk. This is another representation of that network where you can see the models. Very interestingly and expected, you have all the bats here on the bottom interacting mostly with a different set of plants than the hummingbird. And here it's the degree distribution again, showing what I spoke in the past figures, that just a few species do most of the interactions in the system, while a lot of the species interact with very few species on the other level. So some of the advantages of this method is that uses data that it's readily available. You just need occurrence records, an interaction matrix of possible interactions that you can find out doing a good literature review, and also the bioclimatic variables that are readily available on the internet and in several databases. You can use this method to model any type of ecological interaction, and it uses two established methods, the use of distribution models and also network theory to calculate the metrics and how the systems are organized. And it also represents a community level approach to the climate change problem, because I think that looking only to one species or one group of species, it's quite interesting that we know where they may go and and change their distribution, but these species are not alone in time and space, and they need to interact and feed or um, shelter, roost in some other places and spaces. And I think that this is a community level approach that can increase our understanding, and it has a wide application on conservation and climate change mitigation. And some future directions I see for this work is to include a meta network approach where we'll use, instead of pollinators and plants, we'll use the interactions between pollinators and plants, in this case, distributed in different sites. And we can understand which interactions are mostly at risk and where these interactions are at risk as well. Another thing that I find that I think these methods could really increase their applicability is with the inclusion of land use models, because species may move and track their environmental suitability, but they may get there and there is just no real environment for them to use and survive. And another thing is combining functional and phylogenetic traits, so we can also understand how networks may reorganize themselves when two species that haven't interacted before get in touch, but you know these species may actually be able to interact because of their traits, or being just very similar to the species they were interacting before. And this way we could also identify some of the risky traits of species that may be under threat from climate change, for example. So I'd like to thank you all for listening, and copies and the program for funding. All my friends and students at the LEPAI, Laboratório de Ecologia de Paisagens, Jaime and Mariana for all the supervision and talks. I would like to thank Vara and Julia for all the help with coding and programming, and Lucas and Vitor for some of the pictures that they seeded for me to use in the presentation. Thank you.
Obrigada, Thiago. Parabéns pela apresentação. Estamos um pouquinho atrasados no horário, mas acho bom abrir as perguntas agora, se alguém tiver alguma pergunta para o Thiago. Pode ser em português.
Espero que tenha sido claro o suficiente para o pessoal entender, né, no inglês. Tem tempo que eu não falo tanto assim. Estou meio enferrujado.
Se ninguém tiver ninguém tem pergunta, se ninguém tiver, eu tenho então uma perguntinha para você, não sei se eu entendi direito. Então, basicamente, para fazer essas modelagens, né, para ver como essas redes de interações vão mudar como efeito de mudanças climáticas, você utiliza basicamente a distribuição das espécies, assim, cenários de como essa distribuição com a Alô?
Alô?
Fui eu que congelei ou foi o Thiago?
Congelou.
Oi. Alô?
Alô?
Deu para ouvir?
Eu acho que entendi. Eu vou usar... Como é que
Deu
funciona?
Deu para ouvir ou...
Deu para ouvir.
Acho que foi você, já.
Acho que ela caiu. Mas acho que eu entendi. Ela queria saber o que a gente usa, né, para fazer essa modelagem. Como eu falei aqui, a primeira coisa que você precisa fazer é criar uma...
Caí.
Oi, Eugênia.
Estão me ouvindo?
Agora estou ouvindo.
Me ouvindo? Eu não estou ouvindo. Deu para ouvir a minha pergunta ou eu tenho que repetir?
Mais ou menos, cortou no meio.
Alô?
Alô? Eugênia?
Voltei?
Voltou, estou te ouvindo.
Voltei, estão me ouvindo?
Sua imagem está congelando às vezes, mas estou te ouvindo.
Alô?
Oi, Eugênia.
Oi, estão me ouvindo?
Sim.
Sim, sua imagem está congelando às vezes, mas a gente está te ouvindo.
Estão me ouvindo agora? Voltei? Será que eu voltei? De
Voltou.
verdade?
Te ouvindo, te ouvindo. Estou te vendo, mas estou te ouvindo.
Ok. Tomara...
Na hora que ela vai fazer a pergunta, ela cai.
Mas, Thiago, qualquer coisa prossegue na explicação que você estava dando já, se você conseguiu
Então,
pescar mais ou menos.
eu acho que eu entendi mais ou menos. Acho que ela quer entender o que eu uso, né, de entrada nos modelos, né? A primeira coisa que você precisa é criar essa matriz de incidência com as interações possíveis, né? Que a gente vai usar lá na frente para construir as redes locais. E depois a gente vai criar os modelos de distribuição, que de entrada nos modelos você só precisa dos registros de ocorrência e das variáveis climáticas, né? Tanto para o presente quanto para o futuro, nos cenários que você quiser. Existem muito mais do que esses dois cenários, mas eu preferi fazer só no business as usual, né? Como se nada mudasse, e no pior cenário, né? Então, com os modelos de distribuição, você vai calcular a adequabilidade média de cada espécie nesses quadradinhos. E para cada quadradinho, você vai ver todas as espécies que estão ocorrendo nesses quadrados, vai ter a adequabilidade média, e aí você vai terminar com uma lista, né, de espécies para cada gridzinho com um valor de adequabilidade. E aí você vai construir essa segunda matriz, onde você vai multiplicar a adequabilidade de todas as plantas e polinizadores que estão ocorrendo em uma área, e você vai terminar com essa matriz de forças de interações. Só que o problema dessa matriz é que tem algumas interações que você vai registrar aqui que elas nunca foram registradas, que elas são interações impossíveis. Por isso que a gente multiplica essa matriz de interações possíveis com essa matriz das forças para terminar com a nossa matriz final, onde você só vai ter as interações entre espécies que estão co-ocorrendo no mesmo espaço e que têm as interações registradas anteriormente. E a partir disso, a gente vai construir a nossa rede. E aí, dessa rede daqui, a gente vai poder calcular as diversas métricas. E aí a gente faz uma diferença entre o futuro e o presente para ver o quanto que estão mudando e onde no espaço que essas redes estão sofrendo maiores alterações, né?
Então, basicamente, baseado, assim, são modelos que vão prever aonde as espécies estarão com mudanças em variáveis climáticas, né?
Isso,
então, deixa eu perguntar uma coisa, que eu li que tem vários estudos que mostram que, por exemplo, às vezes tem, como o efeito das mudanças climáticas, tem um mismatch entre, por exemplo, o tempo de floração e polinizadores, né? No sentido que vai mudando o tempo de floração quando as plantas vão florescendo, né? Florescendo, se fala.
Isso.
Por causa de mudanças climáticas e especialmente espécies polinizadoras que são migratórias não conseguem mais, né, polinizar as espécies por causa dessas mudanças no tempo de floração. Eu sei que isso foi estudado em ambientes de áreas temperadas. Não sei se isso aqui nos trópicos também é um possível problema. Então, eu queria saber se isso é considerado nos modelos de alguma forma, se é possível, e se isso é relevante também nas áreas tropicais.
Eugênia, isso é um problema, isso daí já está documentado para várias espécies, né? Eu não sei se aqui nos trópicos isso é tão acentuado quanto nas regiões temperadas, né? Porque eu acho que por ser mais estável, né? O mais estável, assim, né? Ter menos variação durante o ano, eu não tenho certeza se muitos polinizadores são migratórios, né? Mas isso daí seria um problema quando se modelasse, por exemplo, ambientes temperados, né? O maior problema, eu acho, é como que você vai medir essa mudança de fenologia para todas as espécies, né? Conforme você vai aumentando os sistemas, esse é aqui a Mata Atlântica, é uma grande quantidade de espécies, né? Eu estou trabalhando com 800 espécies praticamente, né? Então, você tem informação para cada uma dessas espécies de plantas, eu acho que essa seria uma limitação para incorporar essa informação nos modelos. Porém, eu acredito que se essa informação estiver disponível, vão existir formas de utilizar isso daí para refinar os modelos, né? Exatamente como, não sei, teria que quebrar a cabeça um pouco para fazer isso, mas eu acho que não impede também de usar essa informação para refinar os modelos, né?
Mas então, basicamente, na maioria dos casos, as espécies polinizadoras que você usa são espécies que não são que não migram. São espécies.
Eu acho que são todas residentes aqui. Eu acho que não migram. Pelo menos eu sei que os morcegos eles não migram com muita frequência. Os beija-flores eu realmente não tenho certeza. Eu teria que olhar com mais atenção, né?
Ok, obrigada. Não sei se tem mais alguma pergunta.
Eu tenho uma pergunta breve, assim, se der tempo.
Por favor.
Tiago, desculpa eu ter chegado muito atrasado, então peguei talvez metade da sua apresentação, então não sei se a minha pergunta, se você chegou a falar sobre isso. Eu fiquei lembrando muito de um trabalho da Ecology Letters, do Vieira e do Mário Almeida Neto, da Federal de Goiás, que saiu acho que em 2012, 2013, se eu não me engano, em que eles mostram que as redes mais robustas, e aí redes, no caso, bipartidas, né? As mais robustas são as que têm a menor conectância. Exatamente porque uma extinção ela não se propaga ao longo de todo o network, exatamente porque a conectância é baixa. Olhando até para esse seu slide aqui agora, o que... Aí eu não sei porque eu perdi parte da sua apresentação. Você espera alguma mudança nessa comparação, nesse delta presente e futuro? Para o futuro, então, pensando até nesse trabalho do Mário Almeida Neto, de que no futuro a gente teria redes as mais robustas, logo aquelas com menor conectância, exatamente porque as extinções não se propagam em função dessa conectância baixa, ou não?
Eu acho que sim, porque um dos problemas assim, a gente pode esperar que as espécies mais especialistas, elas também vão ter distribuições mais restritas, elas podem vir a perder mais áreas ainda. E aí você vai ficar só com aquelas espécies super generalistas, né? E que vão ter muitas conexões na rede. E isso vai aumentar a nossa conectância. E apesar de você imaginar assim, vai aumentar a conectância, talvez seja melhor, né? A rede está mais conectada, mas o peso de cada espécie que vai sobrar vai ser muito maior. E se por um acaso essa espécie for excluída de uma certa área, você pode ter uma perda de uma grande parte dessas interações, né? Então, uma conectância baixa faz com que você tenha mais redundância dessas interações também, né? Porque você vai acabar tendo espécies que vão interagir só com uma, duas, três espécies na nossa rede. E perdendo essas espécies, você fica na mão só dos generalistas, só daquelas poucas espécies que, como eu mostrei aqui, né, tem o grau, né, o número de interações maiores. E o que acontece? Se você perder isso, você vai perder uma grande parte da rede. Você vai terminar com um monte de planta aqui embaixo sem ter um polinizador, porque todos os outros que também polinizavam essa espécie já foram perdidos anteriormente, né? Por serem mais sensíveis à qualidade do habitat ou à disponibilidade de recursos e etc. e tal, né? Então, eu acho que esse é um padrão que a gente pode observar, né? Um aumento da conectância, porém, uma diminuição da robustez da rede, né? A robustez é outra métrica que eu estou pensando em avaliar também, né, para o futuro e para o presente. Esqueci de colocar aqui, na verdade.
Perfeito. Obrigadíssimo, Thiago. Faz sentido.
Obrigado. Mais alguma pergunta, gente?
Se não tiver nenhuma outra pergunta, bom, sempre podem contatar o Tiago. Senão a gente agora pode passar para a palestra do Brian. Muito obrigada, Tiago.
Muito
Parabéns
obrigado.
pelo seu trabalho.
Muito obrigado.
Agora, então, vamos passar a palavra ao Brian. Então, o Brian Jennings também é um pesquisador associado ao projeto Cap Sprint. Ele fez a graduação na University of California, em zoologia, na área de zoologia, o mestrado e o doutorado na University of Texas, e ele já foi professor visitante do Museu Nacional. Então, hoje, a palestra dele também vai ser em inglês, e o título da palestra é Molecular Perspectives on Vertebrate Diversification in California and Gondwana. Então, obrigada, Brian. Pode fazer o share screen da sua apresentação.
Obrigado, Eugênia, pela introdução. Ok, vou falar em inglês. So today I'm going to talk about some research projects that my graduate students and I have worked on over the years to give you an idea of my research interest. And I'm going to spend some time talking about research I've been doing since I've been in Brazil as well, including mention a little bit about the ongoing project with the COP-based print. My research uses DNA sequences to infer molecular phylogenies so that we can figure out the evolution of species. But when you consider using molecular data to infer phylogenies, it's important to realize that there are actually two types of phylogenies to think about. One is using the gene tree approach, in which let me get my pointer here. The gene tree is what we directly infer using DNA sequence data. And then we use this tree as a proxy of what we call the species tree. The species tree is what we really want to know when we study a group of closely related species. But we usually infer the species tree indirectly using a gene tree approach. So the gene tree basically involves working out the topology, which is the relationships among the sequences, the DNA sequences that you sample from each species. And another aspect of a gene tree that's important are the gene divergence times. These are the times when the sequences, the ancestral sequences, diverged from each other. And so the idea then is that we use this tree to approximate the species tree. And so most studies in phylogenetics have done this approach, and it works generally pretty good. But there are some types of studies in which this approach doesn't work very well. And so we need to use a different approach. And this approach is based on an area of coalescent theory known as the gene tree, species tree theory. And so here is a diagram that gives you an idea of what this theory is about. It's a mathematical theory that's actually generates these probabilistic models of the relationships between the gene trees and the overlying species trees. I don't have time to explain all this today. This is a topic that I cover when I teach a phylogeography course, which I've done many times at the Museo Nacional with Professor Marcela Wexler. So I'm not going to talk about this today. For my postdoc work that I did with Scott Edwards at Harvard, we studied a group of birds from Australia. So this is one of the Gondwanan continents. And so there we are interested in studying the history of two biogeographic barriers. One is the Kimberley Plateau Arnhem Land barrier in the north central part of the continent. And the other barrier is called the Carpentarian barrier, located to the east, this barrier here. And so the group of animals that we studied here is a group called the Australian grass finches. And so here there are two species of long-tailed finches, Pophyla cuticata and Pophyla hecki, and then their relative, the black-throated finch, Pophyla cincta. So in our study, we basically wanted to use DNA sequence data and this coalescent theory to come up with estimates of the divergence times when these species basically came into existence. So that, in other words, the dates of the speciation events. Okay, these are the Australian grass finches that we studied. There's 3 species. The two long-tailed finches, based on their morphology and distribution, and some data from mitochondrial DNA and other characters suggest that they are sister species. So in the species tree, we have them as sister species, cuticata for A and hecki for H. And then their sister relative, the black-throated finch over here, is believed to be outside. So this is the species tree that we're expecting to find using molecular data and analyses. And in particular, we wanted to estimate these divergence times here, the divergence time between the long-tailed finches, which is this time point right here, and the older divergence time here, which is the time of speciation when the lineage leading to the black-throated finches was created. So for the data, we collected 30 nuclear loci, each of which was about 200 to 800 base pairs long. The average is 550 base pairs. We used a technique known as genomic cloning to develop these loci. It's a very labor-intensive method that we don't use anymore. Now you can use next generation DNA sequencing to sequence your organisms and generate many different loci from the genome. So it's much different nowadays. We only sampled 3 birds, 1 per species, because the idea back then was to sample as many different loci, as many different genes as possible, so that we can maximize the statistical power of the analyses. So, you know, if you think about statistics, biostatistics, you want larger sample sizes than smaller sample sizes, you know, everything else equal. And that's the same idea here with these methods. Okay, so the analyses, we used the software POP to infer gene trees for all loci. So we ended up with 30 gene trees, mainly to just, you know, study them and see if the data looked all right. They did. The main part of the analyses that were important to us was to use the special new software developed by Zhiheng Yang to estimate the species tree topology and the 2 speciation times in the grass finch species tree. So that was the main goal of our study there. Okay, here is a snapshot of the results. Going back to these 2 biogeographic barriers in Northern Australia, our multi-locus estimate for the divergence time of the associated with the Kimberley Plateau Arnhem Land barrier is 0.3 million years ago, or in other words, 300,000 years ago is when this presumed barrier acted to sever the gene flow between the populations of long-tailed finches found on either side of the barrier. And over here to the east, with the other barrier, the Carpentarian barrier, we dated this event of divergence to be 0.6 million years ago, or in other words, 600,000 years ago. Okay, now we're going to move from part of Gondwana land up to California in the United States, where I'm from. And I'm going to talk about the research done by my former graduate students for a little bit. This is Sean Riley. He was a master's student with me when I was a professor at Humboldt State University in California. Sean likes snakes a lot, but he actually studied these interesting amphibians over here to the right that have tails. These are called salamanders, and they are pretty diverse in California, especially in the forested regions. And when Sean finished his project, he published his work in molecular ecology, and so it turned out as a very nice project by him. And to give you a quick perspective on where Sean did his study, here's California here on the west coast of the US. This is the state of Oregon right here. So up here in the northwestern coast of the state of California are where the redwood forests are found. So it's a very wet, temperate rainforest found up here in the northwest coast. And it's up there where Sean did his work on this group of salamanders. And here's a close-up map of the distribution of these salamanders. And you can see that there's a lot of geographic variation in the color patterns. And it's even hard to believe that they're all considered to be the same species. It's, was really hard to believe. And that was the motivation for Sean to study this group, to determine how many species of black salamanders there are. That's the question he really wanted to answer. And so Sean focused mainly on these three major populations here, the red, the green, and the blue, to try to figure out if these different color morphs, this gray one here and the red, the black one here and the green, and then this very nice looking yellow spotted black one here in the blue, if these are different species or not. So Sean went to the molecular lab and collected some DNA sequence data. He collected data for 2 mitochondrial genes and also 14 nuclear loci, which he developed using the same kind of genomic library methods that I had used in my postdoc. He collected data from 48 different individuals, sampled fairly evenly from the 3 populations. And to analyze his data, he used this isolation with migration model implemented in the software EMI, which was developed by Jody Hay. This software is a coalescent theory-based software program, which allows you to analyze multi-locus data sets to infer the population divergence time between two populations or species, and also the rates of gene flow, fluxogenico, between the 2 populations. These are the mitochondrial DNA results. This is the mitochondrial tree, and it's color-coded by population. And as you can see, there's really a lot of genetic structure to these salamanders, as suggested by the mitochondrial DNA. And so basically, the two of the color morphs here correspond to colored parts of this tree. The green branches correspond to this population here on the coast. And the blue here corresponds to this one here in the inland region. And then in the north, there is this northern population. One of the unexpected findings is that the northern population, it has 2 different groups of haplotypes or sequences that cluster geographically, this yellow and the red. And that was unexpected because the animals all look the same. They all have this gray color pattern, and yet genetically, they seem to be different in these 2 areas. So that was unexpected, and that's something that will have to be studied in a future study. When Sean analyzed his nuclear DNA, the 14 nuclear genes in the coalescent framework, he analyzed the gene flow rates between each of these major populations. And basically, the measure for gene flow he used is this 2NM measure. And if this 2NM is less than 1, then we consider gene flow to be negligible, which basically means there's been no gene flow between the populations in historic times. So when we look at the data here, we see that the inland population is very isolated genetically from both the northern and coast populations, because the gene flow rates all associated with this inland are all much less than 1. They're almost 0. So the inland population is very isolated genetically. And between the northern and coast, although the gene flow is higher than 1, it's actually still very low. A value of 2 migrants per generation is still very, very low, and probably not enough to stop the northern and coast populations from diverging genetically more and more. So these things are pretty distinct. When Sean looked at the population divergence times, he found that evidence that they had actually diverged a long time ago. When he looked at the northern versus the southern populations, they diverged more than 2 million years ago. So that's quite a long time ago. It's just before the Pleistocene began. And then the coast versus inland was more recent, but still a long time ago. We're talking about 250,000 years ago. So still, these have been diverged for a long time. My other graduate student from Humboldt was Andy Gottschall. Andy studied lizards in the Mojave Desert. This lizard here is called the Mojave fringe-toed lizard. As you can see, they're very cryptic on the sand. Andy ended up publishing his master's thesis in ecology and evolution about his genetics-based project. These fringe-toed lizards are specialized to live on these kind of environments with sand dunes. These are the same kind of sand dunes you see like in the Lençóis Maranhenses in Brazil and up by Natal in Brazil and the Sahara Desert. It's basically wind-blown sand, and that's where you find these lizards. They don't exist anywhere else. So anywhere in the Mojave Desert where you have these sand dunes, you are probably going to see these lizards. One of the key questions in Andy's thesis was, is the Mojave fringe-toed lizard, Uma scoparia, that's this lizard here, is this a different species from its sister species, Uma notata, which is found to the south in Mexico and the very, very southern part of California in the Colorado Desert. So as you can see, these two species, they're morphologically almost identical. There are some differences. You can diagnose them, but it's very subtle. You can barely, most people will have trouble identifying correctly these two species. So Andy wanted to know if it was 2 species or 1. So he took a similar approach that Sean took. He analyzed 14 nuclear loci, which he developed himself using a genomic library approach. He sequenced 93 individuals from Uma scoparia, the target species, and 14 from the sister species, Uma notata. And he used the same isolation with migration model in the program IM to estimate the gene flow and the divergence between these species. And here's a summary of Andy's results. He found that Uma scoparia diverged from the sister species notata about 1 million years ago, so a long time ago. And he found there's been basically no gene flow between these two species. So really basically 0 gene flow. So clearly these are different species, very strong results there. Okay, now I'm gonna come back down to Gondwana where we all are right now. So I came to Brazil, as Eugenia mentioned. I came down here as a professor visitante estrangeiro on a CAPES bolsa. And I came here to first work at the Museu Nacional to study vertebrate diversification, mostly in the Mata Atlântica. There's the Museu Nacional, of course. And when I arrived in 2010, there was no molecular lab at all in the museum. And so I joined forces with 2 professors in the vertebrates department, Professor Marcelo Wexler, right here, who's now the curator of mammals at the museum, as many of you know. And below that is Professor Paulo Buckup, who is the professor and curator of the fish collection at the Museu Nacional. So the 3 of us teamed up. We wrote a grant proposal for the CNPq to fund a new molecular laboratory, and we got the funding, thankfully. And then we took an old room that looked like a bathroom and turned into a nice shiny molecular lab, which was really very functional. And it's really an excellent lab. And since then, many students, postdocs, faculty, including myself, have used the lab to generate our own DNA sequence data that we've since published in many articles. This is an article that we published some years ago on describing the complete mitochondrial genome for a species of hummingbird, or Ppgee flora, found in the Mata Atlantica. And so that's one paper. Another recent paper published there is describing a new species of snake from the Amazon. This project was led by Paulo Roberto Sampaio, who is a doctoral student at the museum. Another really great project I was fortunate to get involved in here in Rio was to collaborate with this professor at Fundão, Professor Francisco Prodacimi. And the two of us, we had this master student here, Igor Costa, who really liked to do computer program, develop bioinformatics software. So Igor, for his master's, he developed his own software program for searching genome sequences, like the complete genome sequence, to extract all the loci that you need to do a phylogeographic or phylogenomic study of your organisms that you're studying. And his research really turned out excellent, and he published it in this prestigious journal, Genome Research, which has like an impact factor of 10. Really nice work that he did. We tested Igor's program using the genomes from our genome, the human genome, and from the chimpanzee genome and the gorilla genome. And we used the program to extract 292 loci, which we then converted to gene trees. And then using this information, we were able to use these coalescence-based software programs to estimate the time of speciation or divergence between when our lineage, the human lineage, separated from the chimpanzee lineage. And we ended up with an estimate of 4 million years ago, which is exactly what all the dozens of studies in the literature all say as well. So we have there's very solid evidence that it's really 4 million years, and Igor's program gave us really nice data to show this same result. And likewise, when we dated the divergence leading to the gorilla lineage, we came up with an estimate of 6.1 million years ago, which again is exactly what we expected a priori. For the Ppgee print project that I'm working on with Professor Eugenia Zandona, I'm collaborating with her and this doctoral student, Igor Souto Santos, who's a student of Paulo Bucup's at the Museo Nacional. Igor is an expert on the fish genus Palestras, a group of fish, 22 species that live in the southeast of Brazil, in the streams and so forth. And so we began a study to basically build on some work that Igor had published 2 years ago in this nice paper that he published, describing the geographic distribution of these fishes down in the like the Costa Verde region of Rio de Janeiro state, so down near Ppgee. And so Igor published this paper, and this is really the foundation of the project that I'm working on now with he and Paulo and Eugenia. And this is a figure taken from Igor's paper. This shows you what these fishes look like. They are a type of guppy. And as we all know, Professor Eugenia does a lot with guppy behavior and evolution. And so this is a really good model system for her and for other researchers at Ppgee. And also Igor is using DNA sequence data to study the evolution of these fishes. So in our project, we are currently sequencing the DNA of several different species of these fishes. And we're currently analyzing those data and are starting to see some interesting results, which I hope to report to all of you sometime soon. So that's all. I'd like to thank Ppgee Print and the Ppgee at Ppgee for hosting me and having me as a researcher there. I'd like to thank Igor for being a great collaborator, and Eugenia, and I'd like to thank Paulo Bucup as well. And thank all of you for your time today.
Thank you, Brian, for your talk. Very interesting. Vou abrir para questões. Alguém tem alguma pergunta para o Brian? Pode fazer em português também. Ninguém? Se ninguém tiver uma pergunta, então eu vou fazer uma perguntinha. Brian, so in your salamander study, so you said those different, so were they different species or different populations? The salamanders with a different color?
Well, when my student began the study, it was all one species with different populations that all looked different from each other. But when he finished the study, he had evidence that suggested that one of the populations is probably its own species. And yeah, exactly. And the other evidence, the results also suggest that the results suggested all three are species, but the inland one is clearly a different species because it hasn't had any genetic contact with the other populations in 250,000 years. So, and they're morphologically very different. They live in a different habitat that's more dry. So they really seem to be on their own evolutionary trajectory, different than the other populations.
But which one was the inland one?
The inland one had the nice yellow spots all over it. It's like a black one with yellow spots all over it. And
so okay.
Go ahead. Sorry.
No, so no, because I was just wondering, like, if there were some with different colors but the same species, if the color was somehow like any sort of adaptation to the environment or not.
That's a really great question. It has not been studied yet, so we don't know. But it's a great question. We don't know. It could be adaptive, the coloration. Or it could be, yeah, it probably is adaptation because these salamanders, they have very large levels of genetic diversity, so it doesn't make sense that genetic drift or there's no bottleneck or anything like that could really reduce them down to a low level of their genetic diversity. So I don't think it's their coloration has to do with anything random like genetic drift, or I think it probably is a result of selection acting locally in the environments where they are. And so these salamanders probably have predators, probably birds. Probably birds really like them. These salamanders are not poisonous like newts and other salamanders. So I think that they do have to worry about predators, and so it could be a case where the selection is selecting for these color patterns.
Okay, thank you. Really interesting. Does anybody else? Alguém tem alguma pergunta para o Brian?
Pode falar em português, se vocês quiserem.
O Brian fala português. Bom, se ninguém tiver nenhuma pergunta, só queria lembrar que ambos, o Thiago e o Brian, ministraram uma disciplina, né, no ano passado para o Ppgee, e o Thiago foi sobre redes, né, interações, né, interaction networks, e o Brian, your class was, what was the title of your class again? That you taught here?
Molecular phylogenetics.
Molecular phylogenetics. Então, se alguém tiver alguma pergunta, alguma questão relacionada a esses temas, sempre pode contatar o Thiago e o Brian. Ok, pessoal. Então, se ninguém tiver mais nenhuma pergunta, eu quero agradecer todo mundo. E vou lembrar que hoje de tarde teremos mais 2 palestras da doutora Luciana e da doutora Carla. às 14 horas, tá? Então, venham participar para assistir as palestras delas também. Mais uma vez, muito obrigada. Não sei se o Bruno quer adicionar alguma coisa antes da gente se despedir e para voltar hoje de tarde. Bruno? Não? Ok. Então, muito obrigada, Thiago e Brian. E a gente se vê daqui a pouquinho, algumas horas, tá? Obrigada. Tchau, tchau. Parou a transmissão, né, Israel?